I hope you can see my screen. So welcome to everybody. My name is Caterina. I'm a senior account manager of Novogene Europe. I will be the host of today's webinar, our first webinar about spatial transcriptomics. And I'm delighted to share that my dear colleague and product marketing manager, Sonny, will be the speaker today, taking us through the webinar, Beginner's Guide to Spatial Transcriptomics. Before that, I would like to briefly introduce our company. So Novogene was founded in 2011 and has been fast growing since then. We are continuously developing our portfolio in the world of next generation sequencing. So today we are able to provide services within very different fields of science, such as agriculture, oncology, infection, disease, or forensic. We are not only spread across different fields of science, but we are actually also spread across the world of location-wise. Novogene is a global company with contact and sequencing facilities in Europe, Asia, and the US. We also have over 2,600 employees and more than 5,600 customers. Here, I would like to highlight two locations. Our first European sequencing center is in UK, in Cambridge, on the left side. This lab is equipped with Illumina technology and our patented intelligent automated sequencing platform called Falcon 2. If you are curious to see how this robot works, you can see on YouTube a video. This automated workflow enables us to decrease the turnaround time, minimize human error, thereby resulting in consistently high quality outcome. And thanks to our supercomputing capacity, we can process large amounts of data. On the right side, and this is something that I'm personally very much excited about, is our new sequencing facility in Germany, specifically in Munich, which opened like 4 months ago. We are gradually launching more and more service types for this lab. So for example, we can do human, plant, animal, whole genome sequencing, RNA sequencing, and pre-made library sequencing, but we have also received the new PacBio review machine there. So every day we just improve and we can offer more and more service from there. In terms of service, we offer a full range of next generation sequencing, So human, plant, and animal whole genome sequencing, various type of RNA sequencing, and various type of microbial sequencing, and also sequencing with epigenomics. Outside this slide, we do also offer oncogene panel for cancer research, and also accept pre-made libraries where we take care of the sequencing part. So why not Novogene? There are many reasons why you should choose Novogene as your sequencing partner. Providing consistently high-quality services is our priority. To achieve that, we perform multiple quality control. So for the samples QC, library QC, and finally the data QC. And we have a large capacity, which we are increasing all the time. Recent example of this is the opening of our new German lab, and also investing in the newest sequencing technologies with improved throughput. Our patented Falcon 2 technology has not only a positive impact on quality and capacity, but also turnaround time, since Falcon 2 can run 24/7. We offer customizable end-to-end solution. This means that we can process your project from the extraction to the analyzed data, but you can also send us extracted DNA or RNA or pre-made libraries and choose to only receive raw data. Our product management team and laboratories are continuously optimizing protocol and services to ensure we have a sequencing solution that fits your need. So in relation to that, we are equipped with a different platform. So Illumina, if you want to do the short reads, and PacBio and Nanopore, if you want to do the long read sequencing. So different platforms fit your needs. Something that I'm happy to share is that we support researchers worldwide. And today we have a clear record in the scientific community. For example, we have been involved in more than 4,000 projects and cited in over 580 publications with a total impact factor more than 4. We are also present with a contact person in many European countries, And you can contact us directly or via our website. We regularly host seminars at the Institute. So if you would like to organize a seminar near you, let us know, because we will be very, very happy. I would like now to introduce our talented European sales team. We are 48 people, and they are based, in different regions across Europe and function as your first person of contact in Novogene. Speaking on behalf of myself and my dear colleagues, feel free to reach out, and we speak your language, so we will be more than happy to help. So now I would like to welcome our speaker, Sonny, our product manager, and enjoy the webinar. Goodbye.
Thank you, Katerina, for the introduction. Good morning and good afternoon, depending on which part of the world you're joining from. Give me a minute to set up my screen. I hope you guys can see my screen now.
Yes, we can. Yeah.
Perfect. Okay, then without further ado, let's get into the topic for today's seminar, which is spatial transcriptomics. Here is a quick overview of what I will discuss in today's seminar. I will start with an introduction to omics technologies and focus on transcriptomics, and then move on to modern day transcriptomics technologies. I will then move on to spatial transcriptomics, and which is the focus of today's seminar. We will take a look at why we need spatial transcriptomics and discuss about the different types of spatial transcriptomics technologies, such as imaging-based or sequencing-based spatial transcriptomics technologies. Then I will move on to our newly launched spatial transcriptomics service, which is powered by the 10x Genomics Visium platform. I will discuss the advantages of this platform over the others, the working principle behind this platform. I will also offer a step-by-step detailed explanation of our service workflow from beginning to end, the sample requirements for this service, and the accompanying data analysis. I hope that by the end of this talk, you will get an overview of this technology, as well as our spatial transcriptomics service workflow, and get an idea of how we are able to support your spatial transcriptomics research. If I say biology is complicated, I hope that most of you will agree with me. Let us take the example of a human biology as an example. On an average, a healthy adult human is composed of 37 trillion cells, and there are about 200 different cell types. Almost every single cell in the body contains a copy of the approximately 3 million DNA base pairs that make up what we know as the human genome, and each cell has its own epigenetic program. Apart from that, there is an enormous complexity within each cell. Each cell has its own unique set of expressed genes in terms of hundreds of thousands of mRNA, and each cell also has a different set of proteins that are being expressed. And as these numbers suggest you that studying biology and biological processes can often be quite complicated. Omics technologies can help us unravel the mystery behind the complexity of different biological processes. And omics refers to the study of totality of something. In the context of biology, omics technologies are employed for probing and analyzing large amount of data representing the structure and the function of a class of biological molecules of a given biological system at a particular level. So this can be either at cell level, tissue level, or at organism level. And this has substantially revolutionized our methodologies used for interrogating biological systems. The outputs of omics technologies are named on a basis of the particular biomolecule level, followed by the suffix omics. So for example, genomics or transcriptomics, where transcriptomics is referring to the study of the total of transcriptomes. And simultaneous study of each of these omics can provide a more accurate, holistic, and representative understanding of the complex molecular mechanisms underpinning biology. Now moving on to transcriptomics. So the transcriptome is the complete set of transcripts in a cell or a group of cells under a particular condition. So this could be a specific developmental stage or physiological condition. Transcriptomics, which is transcripts, plus omics is the study of the transcriptome. And most commonly, this is employed to profile the mRNA content of the cell or the gene expression of the cell. And some of the applications of transcriptome could be for interpreting the functional elements of the genome, revealing the molecular constituents of cells and tissues, and understanding organ development and disease progression. Over the years, several technologies have been developed to study the transcriptome, such as northern blotting, microarrays, or qPCR. RNA-seq or RNA sequencing is one such technology to interrogate the transcriptome that uses next-generation sequencing technologies to not only reveal the presence of RNA, but also the quantity of RNA in a biological sample at a given moment, allowing us to analyze the continuously changing cellular transcriptome. Compared to many of the traditional technologies, such as, for example, microarray, the RNA-seq is not limited to pre-designed probes. It offers higher sensitivity and dynamic range for detection of low abundance transcripts, isoforms, and subtle changes in gene expression. The applications of RNA-seq can be broadly divided into ones that need qualitative information and the others that need quantitative information. So qualitative applications such as genome annotation, orientation of transcripts, determining the boundaries of exon or intron, need information about what are the different types of RNAs that are present in a sample. On the other hand, applications where one needs to know about the differential gene expression level require us to know how many different RNA types are present in the cell and how many of these RNA types are also present in the sample. So RNA-seq is a widely adopted profiling strategies to interrogate the whole transcriptome of tens of thousands of genes. However, the traditional bulk cell RNA-seq approaches suffer from mixing and adding up data from multiple cells. With this, the bulk RNA-seq, we can obtain an average of gene expression profile of all the cells in a sample. However, this has the disadvantage of giving us the impression that all the cells in a sample have the same expression, which is not usually true. The transcriptome of even closely related cells exhibit considerable heterogeneity. This information regarding the heterogeneity of the different cells in a sample in terms of gene expression profile is lost when using bulk RNA-seq, and this is where single-cell RNA-seq is a more suitable option. Let's look at an example of a tumor tissue sample as a case and compare the differences in terms of information or sample processing when comparing bulk RNA-seq to single-cell RNA-seq. So single-cell RNA-seq overcomes the major limitations of bulk cell approach. It allows us to differentiate cell types, analyze the heterogeneous characteristic, including the biological functions of different cell subpopulations and their possible interactions. In terms of sample processing for bulk RNA-seq, the total RNA is extracted, whereas for single-cell RNA-seq, single-cell suspension needs to be obtained. The tissues under study are usually dissociated by physical homogenization to release the individual cells. And depending on the application, one might also need to enrich for specific types of cells. The cell suspension with single cells is then subjected to single cell capture, and there are several different kits available in the market from different vendors. The single cell RNA-seq can give us information about the unique transcriptome of the diverse cells, and it can also provide information about the intrasample heterogeneity in addition to the intersample heterogeneity, which is lacking in case of bulk RNA-seq that only gives us an average gene expression and allows us to compare the average gene expression between two samples. That being said, single-cell RNA-seq is a more expensive approach compared to that of bulk RNA-seq, and therefore one needs to be mindful of whether the research question that they are studying needs this single-cell level of information. Here is a range of our RNA-seq solutions to enable your transcriptomics research. We offer sequencing solutions tailored for different RNA species. For example, mRNA-seq for sequencing of mRNA, or LNC-RNA-seq for long non-coding RNA, a circular RNA-seq, and small RNA-seq. We also offer what we call as the whole transcriptome sequencing option that allows you to profile the different RNAs all together in a sample. We do understand that researchers can often work with with difficult samples from which getting a large amount of RNA is not possible. For example, microdissected tissues or sorted cells or biobank samples. For such cases, we offer low input or ultra low input RNA sequencing option. We also offer support your single cell RNA-seq research. At the moment, we do not offer library preparation services, but we do accept pre-made library that have been prepared from using different kits from different service providers such as 10x Genomics or BD Rhapsody, and are able to offer you sequencing services for your pre-made library that you prepare. We also offer RNA sequencing solutions for microbes and microbiomes in the form of prokaryotic RNA-seq or metatranscriptome profiling. Using the latest Revio from PacBio and Oxford Nanopore, we also support sequencing of full-length RNA. I started out my talk by saying that biology is complex. Just as no man is an island, a cell's behavior and function are not isolated, but rather influenced by neighboring cells organized in a three-dimensional space to form a tissue or organ. Case in point, cancer growth and progression. Cancer growth and progression are spatial processes that involve the breakdown of normal tissue organization, invasion, and metastasis. Studying tumors in a spatial context will advance our understanding of how the complex crosstalk between the cancer and the surrounding non-cancer cells evolve. When it comes to neuroscience, spatial information is important for questions related to disease progression of different neurodegenerative diseases and understanding why certain neurodegenerative diseases affect one part of the brain versus the other, or how do memories develop over time. Spatial organization is also crucial for organogenesis during embryo development. And these are just some of the examples of biological processes where spatial context is relevant. What you see here is a typical tissue sample from a researcher, and the morphology shows how complex and heterogeneous a tissue can be. And what is lying underneath the surface of this complex sample is difficult to unravel. Interrogation of complex heterogeneous tissues require assessment of the spatial architecture. For processes where cellular function needs to be investigated in a spatial context, spatial transcriptomics allows us to profile transcriptomes while preserving spatial information. It helps measure gene activity at a distinct spatial location within a tissue section. Recent spatial transcriptomics methods enable high-resolution characterization of transcriptional patterns and reconstruction of tissue architecture. Spatial transcriptomics was named as the method of the year in 2020 by Nature Methods. And in order for a method to be nominated as the method of the year, it has to fundamentally impact and dramatically advance the way scientists explore questions that are otherwise unanswerable. And this goes on to suggest the importance of spatial transcriptomics in solving complicated biological problems. Investigating cellular function in a spatial context could offer us a more holistic and comprehensive view of these biological systems, can help us reveal novel mechanisms of organ development and diseases, and lead to new strategies for diagnostics and therapy. Here are some of the application of spatial transcriptomics, starting with obtaining a complete view of the tissue architecture to biomarker discovery. And one can also use spatial transcriptomics to obtain spatiotemporal gene expression patterns. Very broadly speaking, there are two ways to profile transcriptomes while preserving spatial information. Firstly, by imaging the mRNAs in situ. by microscopy, and this is what makes the foundation of what we know as the imaging-based spatial transcriptomics technologies. The sequencing-based methods, on the other hand, combine classical microarray technologies with current-day next-generation sequencing technologies to make spatial localization of gene expression possible. I do want to mention that this is a very broad classification of the several spatial transcriptomics technologies into two categories. There are several spatial transcriptomics technologies that use a combination of these or may not be as straightforward to classify in one category versus the other. Let's look into the different options for imaging-based spatial transcriptomics. When imaging mRNAs in situ, there must be a means of distinguishing the different RNAs species. One such imaging-based method is in situ hybridization, or ISH. In this method, we use fluorescently labeled gene-specific probes that can hybridize to the mRNA. Hybridization here is referring to the polymerization of single-stranded mRNAs to single-stranded probes with a complementary sequence. Then we can image the fluorophore locations and repeat this several times to generate images for different genes. The other method is known as the ISS, or the in situ sequencing, in which transcripts are directly sequenced inside a tissue block or tissue section by sequencing via ligation, or the SBL technology. Here, instead of using gene-specific probes, we profile 1 to 2 nucleotide bases at a time of the primed and amplified transcripts that are generated using rolling circle amplification. Each base or the 2-base sequence is linked to a fluorophore and a different fluorophore, enabling the visualization and recording that ultimately leads to the identification of each transcript. The ISH and the ISS methods, which are the imaging-based transcriptomics, are useful because of their high spatial resolution and allows us to profile mRNA at subcellular level. Some of the examples of ISH are merfish, stickfish, and some examples from ISS are StarMap and FISTIC. There are several companies that have commercialized these different spatial transcriptomics methods. For example, merfish is available as a technology is available from Vizgen. Now, moving on to sequencing-based transcriptomics technologies. These technologies require the capture of mRNAs from the tissue, synthesize cDNAs, and then count the gene-specific sequences via NGS. Importantly, positional information is retained at the point of mRNA capture. One such approach of sequencing-based spatial transcriptomics is the array-based method. And in this approach, ligation of the mRNAs to spatially barcoded probes in a microarray is performed. The tissue is mounted over an array such that the released mRNA is captured locally by the spatially barcoded probes, converted into cDNA, and then sequenced using NGS. The other method involves a direct capture and recording of locations, such as via use of microdissection or microfluidics. The genes are barcoded with gene-specific photocleavable probes, for example. When UV light is shown on the tissue, one region of interest at a time, the probes are released, and these are sequenced using NGS. The advantage of using sequencing-based spatial transcriptomics methodologies over imaging-based is that one can avoid using image processing pipelines. The other major advantage is that usually sequencing-based spatial transcriptomics methodologies are untargeted, and thus they can profile the whole transcriptome for any organism. That being said, the spatial resolution and the mRNA recovery rates are typically lower for sequencing-based technologies compared to imaging-based technologies. Now, some examples of array-based methods is the 10x Genomics Visium platform, and we will talk about this in really a lot of details over the next coming slides. At Novogene, we recently launched the Spatial Transcriptomics service, which we offered using the 10x Genomics Visium platform. Now, this platform provides us tissue morphology information via staining and imaging. While FFPE is a common tissue preservation method by the researcher, it damages the RNA, which complicates the transcriptome level investigation, and the 10x Visium spatial gene expression platform overcomes these challenges and is compatible with FFPE blocks or pre-sectioned FFPE tissues on glass slides. It enables discovery of gene expression profiles across tissue sections without the need to select regions of interest. So what do we need to be able to perform spatial transcriptomics using the 10x Genomics Visium platform? First, let's discuss the type of samples that are supported by this technology. So the 10x Genomics Visium can profile a whole transcriptome for any organism that uses polyadenylated mRNA, but this is only true for fresh frozen samples. On the other hand, tissues that are fixed with formalin and embedded in paraffin or FFP tissue blocks, they often have degraded mRNA and thus lack a poly A tail. And gene-specific probe sets for humans and mice from Visium make this technology compatible with FFPE blocks from mouse and humans. And we at Novogene, we are currently offering end-to-end workflow for FFPE blocks from mice and human samples. The next thing we need to be able to spatially resolve the mRNA across a whole mouse or human FFPE tissue section is the transcriptome probe kit. The FFPE transcriptome probe kits are designed to target conserved regions of the protein coding transcriptome of mouse and humans. The probes are also designed to be specific for their target transcript. The Visium probe kit is a split probe kit consisting of split probe pairs, and these pairs are called as the left-hand side probe or the LHS probe, or the right-hand side or the RHS probe. And the LHS or the left-hand side has partial read-to sequence, whereas the RHS has the synthetic poly-A sequence. And I will come to the relevance of these parts soon. For humans, roughly 18,000 genes are targeted, whereas for mouse, almost 20,000 genes are targeted. What is not included in the transcriptome probe kits are mitochondrial ribosomes and ribosomal genes, or immune genes that undergo somatic rearrangements, genes with less than 50 base pairs of coding sequence. The next important component of a spatial transcriptomics experiment using 10x Visium is the Visium spatial gene expression slide with barcoded capture oligos. Now, here is what the slide looks like. It has two capture areas, and there are two versions of this slide. One is where the capture areas are 6.5 millimeter times 6.5 millimeter. They're also available in 11 millimeter times 11 millimeter. Each of these capture areas are identical to each other, and each capture area contains about 5,000 barcoded spots. Each spot is 55 micron in diameter, and the distance between two spots is 100 micron, which is the center-to-center distance between the spots. The 55 micron spot size is what gives Visium its high resolution. Now, depending on the tissue type, about 1 to 10 cells would land on each spot, and this depends on the tissue thickness and the tissue type. Each of these spots is printed with millions of oligos that serve as a mechanism for capturing and barcoding RNA molecules. To a workflow I will get into soon. And here you can see a cartoon of one of these oligos, which contains the spatial barcode, the unique molecular identifier, and the poly-DT, which will be used to capture the transcriptome probes with the synthetic poly-A. Let's look into the RNA capture principle that is employed by this technology. So this capture principle is unique for samples, FFP samples with degraded RNA that typically lack poly-A tail. The Visium CytAssist spatial gene expression slides utilize a new and specialized chemistry that we refer to as the RNA templated ligation method that allows for sensitive and specific RNA detection in FFP samples. The mouse and the human transcriptome probes are split into LHS and the RHS sites, or the left hand and the right hand side. And these hybridize to the target mRNA. And once they find their correct mRNA, they ligate via a ligase. And using the advantage of using the split two probe chemistry is that it reduces non-specific signal over using a single probe chemistry. The introduced poly-A tail here allows for the capture via the poly-T on the slide surface. Another important component of this experiment is the CytAssist platform, which is a small instrument that facilitates the transfer of the transcriptomic probes from the glass slides onto the capture area on the Visium CytAssist slide. Given that we are doing spatial transcriptomics, retaining the correct spatial information is very important, and the CytAssist minimizes mislocalization and maintains spatiality. Let's go now through the workflow stepwise. So the workflow starts with imaging the tissues for cell morphology visualization. This step involves preparation of the glass slides with tissue sections, which is followed by deparaffinization, staining, and imaging. The FFP tissue is then decross-linked so that the RNA molecules are accessible again. The probe panel consisting of the split probe pairs are then added to the decross-linked tissues. Now here you can see the two components of the probe pair, the two sides, and they find the target mRNA, and if both of the probe pairs are complementary to their target RNA, they will hybridize. Once the probe pairs have hybridized, they will be ligated together to form the full-length ligation product. The ligated product has the read2S sequence and the poly-A tail, which will then be released from the target RNA using RNase treatment. So upon RNase treatment, the mRNA, target mRNA is degraded, and we have this single-stranded ligated DNA probe with the synthetic poly-A tail. This is followed by tissue permeabilization, which will allow the full-length ligation product to be captured in the capture area. And here you can see that the poly-A tailed full-length ligation product has bound to the capture oligo on the slide via the poly-DT. And once that has occurred, we can do a probe extension to incorporate the spatial information, the unique molecular identifier, and the read one into our molecule. And this is done by the probe extension step. Once that step is done, we are ready to take the molecule off the array or off the slide into a tube and proceed with library preparation. The spatially barcoded ligated DNA is amplified via PCR to generate sufficient DNA for sequencing library construction. This is then followed by sample indexing. During the sample indexing step, we introduce I5 and the I7 indexes, and the P5 and the P7 handles, which are necessary for the molecule to bind to the flow cell. And this is the final sequencing library that is ready to be sequenced. This is the final library structure. It begins with the P5 and ends with P7, which are necessary for binding the DNA to the Illumina flow cell. The I5 and the I7 sample indices, which were introduced during the sample indexing. We also see the 10x spatial barcode. This gives us spatial information and allows us to map all the reads back to a specific location in the tissue. The UMI, or the unique molecular identifier, this will be used to count ligation events and to keep track of the number of RNA. The poly-DT tail, this was used for capturing the poly-A tailed ligated probe. The read1 sequence here is used for sequencing the spatial barcode and the unique molecular identifier, which is 28 base pairs in total. The read2S allows for sequencing of 50 bases, which is the ligated probe insert. In general, it is recommended to sequence 25,000 read pairs per spot to be able to get a generous amount of gene detection rates. That being said, the sequencing depth is often influenced by factors such as the coverage area of the captured region on the slide, the sample type, and the complexity. So here is the schematic of the workflow starting from sample preparation and imaging all the way to data processing and visualization. And I understand that it might appear the whole process is quite complicated with quite a few steps in between, but do not worry, our transcriptomic service supports this entire process from beginning to end. All you have to do is send us your FFP samples and we will provide you data complete with analysis. While we are on the topic of spatial transcriptomics using 10x genomics, this month 10x announced its latest Visium HD, which is a higher resolution version compared to the technique that I described today. And you can see the difference in resolution by comparing these two different images. The workflow is exactly the same. What's different is the capture area. The capture area has 11 million squares compared to the 5,000 spots that I described earlier. This allows for a continuous coverage, and that's why you do not see the spots that you see in Visium V2. Now, in terms of sample requirements, as I mentioned earlier, it is currently available for human and mouse samples only, and this is because the transcriptome probe panel is designed against human and mouse genes. The FFPE blocks, it is better if they are stored for less than 3 years, and we prefer to have FFPE blocks compared to FFPE sections to be able to obtain better results. And when transporting FFPE samples, make sure to include ice packs or dry ice to prevent any chance of melting of the blocks during transportation due to higher temperatures. Now, what about the RNA quality in these FFPE samples? So typically, when determining RNA quality in FFPE samples, a common parameter to measure is the DV200, which is the fragment distribution value of RNA fragments that are larger than 200 nucleotide. For the spatial transcriptomic service for FFPE samples, the samples need to have a DV200 greater than 30% to be used for library construction. Lower DV200 in a sample can significantly impact library sensitivity. And typically, we will take a small number of paraffin sections and use them for RNA extraction and then determine the DV200 quality index using the fragment analyzer for Agilent. If you have access to such an instrument, you can go ahead and determine the RNA quality before sending us your sample for further processing. Now, moving on to the last part of the seminar, which is about data analysis. So what does the Visium data look like? First of all, we have the bright field image of the hematoxylin and eosine stained tissue section. This is a very important part of the data set because here you will have access to detailed morphological information at very high resolution. The other part of the data set is the gene expression matrix. These represent the dots where we have captured mRNA. Each spot represents an average expression of a mixture of cells. And the Visium data is the overlay of the tissue image with the gene expression matrix. And as I mentioned before, the resolution here is about 1 to 10 cells per spot, but this varies depending on the tissue type. So when you have performed the Visium experiment, you will have the raw sequencing data from the Illumina machine, for example, and then you will process this data to get the gene expression matrix that you can work with for further data analysis. And this can be done using the Space Ranger pipeline. This is the software provided by 10x Genomics, and it takes the raw sequencing data in FASTQ format and the H&D, the hematoxylin and eosine stained images of the tissue slides as an input. The Space Ranger analysis pipeline aligns the gene expression matrix with the image. It does so by assigning each detected gene transcript captured during the workflow to a spatial location on the tissue image based on the associated spatial barcode. It also generates this nice report that you see here on the right. If you have used the Cell Ranger for single-cell RNA-seq data analysis, you are probably familiar with this. Here you get some useful quality matrix telling you, for example, how well the sequencing went, the mapping to the reference genome. You also get information about how many genes you have per spot. This is a very useful report to assess the quality of your sample. This report also flags if something looks really odd or different. For example, if you have a very few reads mapping to the reference genome, it will tell you that something looks strange. This is also a quick way to check if you have aligned the correct FASTQ file with the correct image. If you see a lot of weird higher transcriptome number in certain regions than you expect, that can be a quick way to figure out if something went wrong during your experiment or during the analysis. Then there is another tab on this report, which is the gene expression tab. And this part can be quite useful as a sanity check. Here you will get information about some basic analysis. You will get spatial visualization of the transcripts on the tissue. You will also get unsupervised clustering result. It can serve as a quality check to know if the clusters represent the tissue heterogeneity and to check if you actually get a lot of transcripts in the regions than where you expect. Now, here is our standard bioinformatics analysis workflow. Our standard pipeline includes both the Space Ranger analysis report that I mentioned earlier, but we also include the SORAT analysis, which is one of the standard analysis pipeline for spatial transcriptomics. We offer analysis of differentially expressed genes and spatially differentially expressed genes. And once such a list of differentially expressed genes has been generated, we are also assigning biological functions to these differentially expressed genes using some standard databases such as the Gene Ontology Database or the GEO enrichment or KEGG or Reactome enrichment, for example. So with that, I will summarize my talk by comparing the different transcriptomics methods that one uses currently. So I'm comparing here spatial transcriptomics against bulk RNA-seq and single-cell RNA-seq. So one can think of bulk RNA-seq as a fruit smoothie where you get an average expression level, but you have no idea about the different fruits that make up the smoothie. However, this method is still very widely used and very useful when it comes to doing comparative transcriptomics and also particularly useful for homogeneous systems. When it comes to single-cell RNA-seq, here you can get information about individual fruits that are present in your sample or individual cells that are present in your sample. You're able to separate populations, define heterogeneity and cell population dynamics. In comparison, spatial transcriptomics, you can think of this as more of a fruit tart, where you can get information about the complete view of the tissue architecture. You know how the different tissues are organized with respect to each other, or how the different cells in a tissue are organized with respect to each other. You can also get information about the spatiotemporal gene expression pattern and analysis using this method. So you might wonder which one of these different transcriptomics methods should you go for? And that really comes down to the level of information your research question requires. And that's something to think about when picking the most appropriate most appropriate transcriptomics technology. Yeah, so with that, I'm happy to take any questions you might have for me.
Thank you, Sonny. Thank you very much for all the information about this product. So we have like a lot of questions, so I think that we will not have much time to do all, but we will start, okay? So the first one is, how do I know if my gene of interest is included in the Transcriptome probe kit?
Yeah, that's a very good question. First, let me just mention the genes that are not included, which are the ribosomal genes or TCR genes or mitochondrial ribosomal genes. or genes that are less than 50 base pairs, and also like non-coding RNA, these are not included. That being said, if you're interested in a specific gene, there is a list of genes that 10x provides on their website, summarizing all the different probes that they include. So you can go through it. I would also encourage you to reach out to us and with this question with your specific gene, and we can also help you with the same.
Thank you. The next question is the transcriptome probe kit same for the new Vision HD?
Yes, it is the same for FFP samples, yeah.
Okay. So if there is a mutation within the binding regions of two probes, will the probe still bind to the mRNA?
So according to 10x, the technique allows for mutations to be there. So if there is one mutation, it is all right. However, that being said, if the mutation is at the junction of the two probes, that might lead to decrease in efficiency.
So will the transcriptome probe sequences work on non-human primate samples?
So the probes for humans are designed against the human genome. So it hasn't been tested against non-human primate samples. So I wouldn't recommend the probe kit for non-human primates.
Okay, so this is connected to the next one because is the vision-based method also established for plant material?
It's okay. So I do want to clear some confusion if there are any. The Visium technology for fresh frozen samples work for any species with poly A mRNA. However, for FFPE samples that require transcriptome probe kits, that's only for humans and mice. So yeah, if you want to study plants, you can do it with fresh frozen samples.
Great, thank you. Do you offer Vision HD?
Not at the moment. That being said, we are, as a company that believes in pushing boundaries, we are always updating our services and keeping track of the latest technologies. This is a fairly newly announced technology from 10x. So keep updated. We will release the announcement as soon as we make those changes.
Yeah, just contact your local account manager and we will
definitely
keep you posted. Can I use custom probes?
So technically, yes, you can design your custom probes, and there is a list of recommendations from 10x about how to go about designing these probes in terms of the GC content or what kind of adapters to add to your probes. We don't typically offer this, but that being said, please reach out so we can discuss your needs and see what can be done for your project.
Okay. And then what is the standard of sample QC? If I fail once, how many chances do I have later on?
So the sample QC would be first is to do an H&D stain image. And once we do this, we provide it to you. You can, of course, do this yourself and get an idea about the sample. So for example, is your sample heterogeneous? Is there a specific region you are interested in capturing? The next part of the sample QC is to perform the RNA QC. And typically we do this for DV200. We would take a couple of sections from the tissue and then perform the RNA QC on it. If you have access to the fragment analyzer, do this in advance to sending us the samples. If you are unsure, you can send us multiple samples. We can do the sample QC, reach out to you, let you know which ones of them passed, and we can proceed with those. The other step in the process could be while we transfer these samples from the glass slide to the capture area, there can be detachments, and this happens usually if the FFP sample was not correctly prepared. So there's something to keep in mind before you send us the sample.
Great. Thank you so much. Unfortunately, the time it's over. So I want to thank Sony, and I want to thank you all of you for your time today. We hope you enjoyed the webinar and found it useful. Thanks for all your questions. As I mentioned, if we haven't managed to answer to your question live, we will get back to you via email next week with answer. We will email you a link to the webinar recording and the slide deck. You can expect to receive this on Friday, and when the webinar finishes, a survey will pop up. So we would really appreciate if you can take the time to complete this, as it helps ensure make future webinars even more relevant to you and your research goals. As a token of our appreciation for your attendance, Novogene is pleased to offer you a 15% discount on your upcoming project. An email will be sent to you within the next 3 days with a code that you can use to apply to the discount. There will be a link in the follow-up email for you to view a certificate of attendance if you need one, and these can be printed from your browser window. In the meantime, you can follow us on Twitter, LinkedIn to keep up to date with future webinars, offers, and new. We're looking forward to see you all in the next webinar, and I say you goodbye. Thank you very much.